My name's Skylar, and today we'll be talking about prokaryotic DNA replication. Starting out with the initiation step of DNA replication, I chose to demonstrate using pipe cleaners and beads. First, the origin of replication is established. Second, helicase unwinds the DNA. Then, RNA primase, or the pink heart beads in this case, lays down the primers, or the green pipe cleaners, for DNA polymerase III to come in and begin elongation. Gyrase is not depicted, but it helps to relieve any tension due to supercoiling formation that happens when helicase unwinds the parent strands. Also not pictured is the single-stranded binding protein that binds to the newly separated individual DNA strands. It keeps the strands separated by holding them in place so that each strand can serve as a template for new DNA synthesis. DNA polymerase III, a holoenzyme represented by the big yellow beads, comes in and starts DNA replication, and can only add to the 3' end. DNA is read in the 3' to 5' direction on the parent strand, and nucleotides are synthesized in the 5' to 3' direction. However, one of the parent strands of the DNA is 3' to 5', while the other is 5' to 3'. The pink parent strand is 5' to 3', while the blue parent strand is 3' to 5' here. The arrows in the picture represent the direction of replication. Replication occurs in opposite directions on either strand. The leading strand is synthesized continuously, only requiring one primer, and the lagging strand is synthesized in a series of short fragments known as Okazaki fragments, consequently requiring many primers. You can see here the newly replicated anti-parallel strand in orange. When DNA polymerase III runs into the RNA primer, it detaches and is replaced by DNA polymerase I, represented by the green bead, and performs a 5' to 3' exonuclease activity on the parent strand. This exonuclease degrades the primer and synthesizes DNA in its place. We see here the new replicated sections in orange after DNA polymerase I replicates in the 5' to 3' direction. Once the RNA primer is removed, the two sections are joined by DNA ligase, represented by the dolphin beads on the right. When the two replication forks meet each other, replication stops and two new DNA strands are joined by DNA ligase. Note that the colors here are not significant. Just know that DNA replication is semi-conservative, meaning that one strand comes from the parent strand and one is a new strand. If there were no DNA polymerase I exonuclease activity, DNA replication would not happen correctly. The RNA primers would be left behind, and the Okazaki fragments would be left as is. Therefore, this would likely increase genetic mutations, as there would be no proofreading component to correct for mistakes.